I would like to explain the polymerase chain reactions, PCR. PCR is a in vitro technique that takes specific sequence of DNA of small amount, amplifies it and produce millions of copies. So it can be compared with photocopier machine or Xerox machine. So you know the PCR can make millions of copies, and the same manner the photocopier or Xerox machine also makes the number of photocopies. So every office we have a photocopier. In the same manner, every molecular biology laboratory or clinical laboratory or any research laboratory must have a PCR machine. This PCR is discovered by Carey Mullis in 1983. So he also won Nobel Prize in Chemistry for PCR discovery. So PCR is widely used in medical sciences for detecting many sexually transmitted diseases, include HIV, gonorrhea, chlamydia infection, etc. So not only the sexually transmitted diseases, it is also important for genetic predisposing for defects such as factor V Leiden. This factor, clotting factor, specific gene mutation that results in thrombophilia. So this PCR is used not only in medical sciences, it's also used in forensic sciences, in the law of enforcement, and also genetic testing of animal stocks in animal sciences and agriculture sciences in the case of vegetable hybrids, and also in pharmaceutical sciences like drug screening process. So same manner, the Xerox machine or photocopier need some items or requirements for making photocopies such as paper, ink, etc. Same manner, the PCR also need some items in order to make the millions of copies of DNA of interest. So mainly we need some components for making the number of copies of DNA sample. So first of all, we need a source DNA sequence of target region must be known. So first of all, we need a DNA sequence of interest, and second is primers. So primers are typically 20 to 30 bases in size. Simply speaking, primers are small DNA piece. It has reverse and forward primers. So these primers can be readily produced by commercial companies and can also prepared using the DNA synthesizer. So this primer will initiate the DNA synthesis at 3 days end. Then another is the thermostable DNA polymerase. So we are using the thermostable DNA polymerase for making the DNA copies. Example, Taq DNA polymerase. This polymerase is isolated from Thermus aquaticum bacteria, which is not inactivated by heating at 95 degrees Celsius. The another famous DNA polymerase is PFTU, that is isolated from Pyrococcus furiosus, so which is widely used because it is higher accuracy of adding complementary nucleotides. So in olden days, we used native Clenov E. coli DNA polymerase, which is stable only at 30 degrees Celsius. So due to this instability in the temperature, we need to add this E. coli Clenov DNA polymerase at each and every step of the PCR cycle. So that is the main disadvantages of the native Clenov E. coli DNA polymerase, since it is not thermal stable. So we use thermostable DNA polymerase as one of the main component in the PCR reaction. The next is the DNA thermal cycler. So it's a machine which can be programmed to carry out heating and cooling of samples over a number of cycles. Then another component is DNTPs. So this deoxynucleotide triphosphates are the building blocks for new DNA strand. So it includes adenine, guanine, cytosine, thymine, or uracil. Then magnesium chloride. So these magnesium ions act as a cofactor for DNA polymerase enzyme for synthesis of new DNA strand. So finally, the buffer, the pH is around 8.3 to 8.5. So these are the basic components of PCR reaction. Then the conditions of PCR reactions are denaturation of double stranded DNA template, then annealing of primers, then extension of double stranded DNA molecules. So these are the steps involved in the PCR. So first step is the denaturation, followed by annealing and extension of double stranded DNA molecules. Now we will see each and every step. So first step is the denaturation step. This step is carried out at 94 degrees Celsius. With this high temperature, 94 degrees Celsius, the double stranded DNA melts and it is getting broken into single stranded DNA molecules. So this is happen when we increase the temperature around 94 degrees Celsius in the thermal cycler machine. So during this time, the bond between two strands getting broken due to the cleavage of triple band of guanine and cytosine and double bond of thymine and adenine. So this bond, the hydrogen bond, will getting broken due to the increase of temperature. So this broken of double band into two single stranded DNA is called denaturation step. The next step is the annealing process. So annealing of DNA with the primer will take place at 50 to 70 degrees Celsius. So most probably we use around 60 degrees Celsius. So during this temperature, the primers bind to their complementary sequence. So normally the binding or attachment will take place at the 3 days end of the complementary DNA. So the forward and reverse primer will attach with the complementary DNA molecule. So this process is called as hybridization. So the hybridization takes place based on the specific DNA sequence. That means the primers will have a specific DNA sequence containing base pairs that is only specific or particular for the complementary DNA. So during this process, the binding will take place between the base pairs. So for example, the forward primer having the guanine, so it will anneal or attach with the cytosine of complementary DNA sequence. So you can see the thymine will attach with the adenine. So the primer containing guanine will attach with the complementary DNA of cytosine. So likewise, the forward and reverse primer will hybridize with the DNA strand. This is called as annealing process. So the final step is the extension or elongation, or we can say synthesis of new DNA molecules. So at this stage, the temperature is 72 degrees Celsius. Again, we need to rise the temperature. The total process of extension will take place between 0.5 to 3 minutes. So during this stage, DNA polymerase binds to the annealed primers and extends DNA at 3 days end of the chain. You can see this image showing the elongation of DNA primers will happening in the picture. So in this case, the primer will attached already during the annealing process. So there is a Thermus aquaticus DNA polymerase enzyme. It is helping the primer elongation or extension process. So during this stage, there will be an addition of DNDPs, nucleotide bases of the DNA fragment. So during this addition, this DNA polymerase will help the attachment of DNTPs with the complementary DNA sequence. So you can see this thymine DNTP is added and attached with the adenine of complementary DNA of interest. So then next the guanine is attached with the cytosine of this complementary DNA sequence. So accordingly the primer extend or elongated until the new DNA fragment synthesized. So in the same manner the reverse primer also will extend the primer addition, DNTP's addition and attach with the complementary DNA sequence with the help of DNA polymerase enzyme. So until the new DNA strand has been synthesized. So this is the extension process. Once the new DNA strand has been synthesized, the DNA fragment is getting doubled. So at the end of the first cycle, we may have a 4 DNA molecules. So this is the figure showing the different cycle of PCR machine. So this is the source DNA, 2 double stranded, that means 2 single stranded DNA are there. So during the first cycle it is getting double. There is a 4 copies produced with the first cycle. So in the second cycle there is a 8 copies produced. So the 4 become doubled, it's getting 8 copies of the DNA. So we may increase the cycle, we may run the cycle even more than 30 cycles. At the end of the cycle, 30 or 35 cycle, we may have millions of copies of the source DNA of interest. So during the PCR or after completing the extension or synthesis of DNA molecules, we need to take the F&R tube containing samples. We need to load into the electrophoresis gel system. So after loading into the electrophoresis, the separation will take place in the electrophoresis process. So when the electrophoresis process ends, we can go for gel documentation system in order to visualize the synthesized DNA fragment. When you look at the synthesized DNA, if it is not work, if no product is produced, I would like to give some tips in order to improve the PCR work perfectly. So first case, we may check the DNA quality. That means the source DNA quality should be rechecked if the product is not found. Moreover, we may reduce the annealing temperature, or we can increase the magnesium ion concentration, or we may add some DMSO to the assay, dimethyl sulfoxide to the assay, or we may use some different thermostable enzyme. Instead of Thermus aquaticus, we may go with other enzymes, or we may use the new stocks instead of using the primer old stocks. If there is any extra spurious product band found, if the DNA band is excess or the size is more or some extra band are produced, by the time we may have some tips. For example, we may increase the annealing temperature, or you can reduce the magnesium ion concentration, or reduce the number of cycles, or we may go with different enzymes. So this is the example of PCR program. So initial denaturation should be 95 degrees Celsius for 5 minutes. For 30 cycles, we may have denaturation with 95 degrees Celsius for 30 seconds, annealing 55 degrees Celsius for 30 seconds, extension 72 degrees Celsius for 45 seconds. Finally, the final extension or holding time should be 5 minutes with the same 72 degrees Celsius. So advantages of PCR. So advantages of PCR, that includes a small amount of DNA. Since we need a only small amount of DNA for PCR, then the results obtained will be more quickly. Within a day, we may get the PCR results. Then another is we are not using the radioisotope. So there is no radiation hazard during the PCR process. So PCR is much more precise in determining the size of alleles in the case of essential for some disorders, genetic disorders. So PCR can be used to detect point mutation. So these are the few advantages of PCR. There are many advantages over other diagnostic techniques. Then finally, applications of PCR. So applications, we have a many abundant PCR uses. So this is a few. For example, we may detect some bacterial disease such as gonorrhea or TB, some other diseases like chlamydia diseases, viral diseases like HIV. We may detect some genetic disorder or mutation disorders like factor V, Leiden, or we may go with some forensic testing. There are many more applications.